The Sustainable Development Goals are the United Nations initiative to change the world. They will guide development programs in all countries for the next 13 years. If these Sustainable Development Goals are reached, we will live in a much better world by the time we turn 30. It is our generation's turn to save the world. For this, we need a shared vision, an all-inclusive framework for action, and clearly defined goals. Goal 3 out of the 17 Sustainable Development Goals is to ensure healthy lives and promote the well-being for all. Though significant strides have been made in increasing life expectancy and reducing some of the common killers associated with child and maternal mortality, many more efforts are needed to fully eradicate a wide range of diseases. Cancer remains one of the most feared diseases in the modern world. According to the World Health Organization, it affects 1 person in 3 and caused a quarter of all deaths during the year 2014. In the year 2015, 9 million people died of cancer. Over the next 2 decades, the number of cancer cases is expected to rise by over 70%. One of the major causes of cancer is its late detection. In most cases, cancer is detected at the 3rd or the 4th stage, after which it is usually incurable, leaving medical experts helpless and cancer patients devastated. Due to the varying DNA of each and every person, there are infinitely many DNA combinations which can lead to the formation of cancer. According to a recent study done by the University of Leeds, there are more types of cancers than there are people in the world. With this consideration, we thought of making a device which is endowed with sufficient features to detect cancer in its early stages through a very simple procedure which does not need expert supervision. The device will take a blood sample from a person's fingertip and will then analyze their DNA. It will also analyze the proper functioning of the oncogenes and the tumor suppressors. If there is even a slight change in the shape of the cell which has been formed by the oncogenes, the person will be notified that they are at a potential risk of cancer or that cancer has already become malignant in their body. The device will provide more accurate results as a molecular assay capable of reporting microRNA presence in biofluid-derived RNA samples will be installed in the device. In presence of the target microRNA, the assay generates a fluorescent signal that is visible to the naked eye. The reaction takes place in isothermal conditions, effectively simplifying the microRNA detection setup. The detection limit of the assay is within the dynamic range of circulating microRNAs in biofluids. We further propose to link this device to doctors, hospitals, clinics, and even search engines to provide the patients with quality expert advice and treatment steps. The device will offer 2 options, one which is free of cost and the other the expert advice which would come at a cost. Early prognosis will help millions of individuals around the world in detection of this disease. This would greatly increase the chances for successful treatment. Our goal is to make this an effective prognosis tool which helps to decrease mortality and improve survival rates by detecting cancer at an early stage. Let us come together to achieve this. This is our chance to shape the world that we want.